Hi, folks. I'm Brad Shaffer at UCLA, and this is a talk with Peter Scott, very much in the same theme as the last two talks. In many ways, I think we probably should have just done a JJ, Peter, and Brad joint talk on building genomic resources to guide repatriation of illegally traded turtles and tortoises. And because some of the stuff that I'm planning on talking about, JJ's covered. I'll try and move through some of it quickly and be able to move on to some more detailed stuff on a couple of specifics in a moment. Sorry. So I want to start with two quick facts that are important, I think, for turtle conservation genomics. The first is that we are building a very important and very useful set of reference genomes for turtles globally, and that's really important. And the second is that genomics of any sort is actually becoming affordable, and I'll talk more about that a little later. So in terms of those reference genomes, this is a phylogenetic tree, a circular tree for turtles from a paper that we have under review right now that Bob Thompson's the lead on. He'll be talking about it in a later session of the TSA meetings this year. And with some very generous financial support from Tim Gregory, we now have a well-established reference genome, the kind of thing that Peter was talking about for Terrapini triunguis, for every one of the 14 families of turtles. And that's just a really important resource. It's a great starting point for the kind of work that JJ, Peter, and I are all talking about. And just keep that in mind. We have that for geomydids, testudinids, trianichids, the whole gamut of turtles. Okay, so confiscations of turtles have been a big issue in the world, and Craig talked about this, JJ talked about it. In a nutshell, they happen regularly. In my view, at least, they seem to be happening more regularly. There are big US confiscations at LAX, Los Angeles International Airport, for example, right down the street from me, at marketplaces across the United States. Internationally, this is a picture of the big Siberoculiotensis confiscation a few years ago. We heard about radiated tortoises, the big one in Mexico. It's a problem. And as JJ pointed out, we need to think about not just those confiscations, but also what to do with those turtles and tortoises and how to put them back on the landscape. And so what I want to talk about today, as JJ did, is using genetics to aid in those repatriation efforts, but also recognize that it's not just the repatriation efforts, it's also identifying where those illegal animals came from. And so if you're wanting to focus, policing efforts on the illegal trade, by knowing where they came from, even if we can never put them back, it'll help us focus on where attention to stop this illegal trade might focus more. So let's just think about this in a bit more detail. So this is actually it's a picture of a diamondback terrapin in the state of Florida. And this is actually a project that Peter and I are engaging with right now with the state of Florida. So say you wanted to be able to do this kind of exercise of assembling a sort of genomic resource for terrapins in the state of Florida. What would you do? Well, the first thing you might do is identify a series of sites, something like that, that span the state of Florida where diamondback terrapins are found. Using some genetic method, you would sequence each one of those, a turtle from each one of those dots, and you only have to do one at some level of depth and coverage. And then if you ever had another animal that was, say, a confiscated animal or an animal that showed up in an animal rescue center and you wanted to know where to put it back, you would sequence that animal for some part of its genome and compare it to all the dots. And whoever it was most closely related to, you could say, well, at least it came from somewhere in that region. And that's the basic goal of the exercise. I think as we think about this exercise, it's important to think about what genetics can do for us and what it can't. So it certainly can identify the closest relative. You know, if we go back to that picture, it can say which of those red dots that target animal is most closely related to, with some degree of precision, as Peter pointed out. It may be able to also focus in on genes that are important in local adaptation. For example, with diamondback terrapins, there is some evidence that there are genes that contribute to the ability to tolerate salt water. And if some areas have a greater genetic propensity to be able to do that than others, then we might be able to say, okay, well, our animal came from a population that's very salt adapted, and think about putting it back there. So there's some of those kinds of ecological issues that we can also work with. What it can't do generally is pinpoint the precise populations of origin. So it can't say where in between those red dots we go. It should go exactly. It can only do it at the level of sort of resolution that the red dots give us. And that's important to remember. Another important point in this thing to think about in this issue and in this process, and Peter mentioned this a bit, is how much data have for each reference animal. And you've got to keep straight, there's a difference between what we need for the reference animals and what we need for the animals we're comparing to those references. And here, focusing on the reference animals, there's really two alternatives. One of them is to sequence a part of the genome. It might be a little part, like one mitochondrial gene. It might be a bigger part, like RAD-seq or the target capture approach that JJ mentioned. The reason to use a part of the genome is it doesn't cost as much for the reference animals, okay? The price you pay for that is that you don't get as much resolution and as much information as you might. The second alternative is to sequence the entire genome and for the reference animals. And as I'll mention in a moment, that is more broadly useful, but it costs more. Okay, so that's the trade-off. So let's think about this a little bit more. And for the first alternative, which is using a limited part of the genome for those georeference samples, for those reference animals. So here's just sort of an example using, for example, mitochondrial DNA, which is essentially what DNA barcoders do. And you might think about here, the sort of reference animal is this solid black line going down the middle of this plot. So there's a reference genome for cytochrome B. And if you have confiscated animals, you can only use the information that's present in that cytochrome B. You have to sequence the same piece of cytochrome B in the confiscated animals as you do in the reference animals. And then you can compare it back, and whatever the resolution of that gene or that collection of genes is, will limit the resolution of what you can say. So going forward, once you have that set of reference genomes, you can only use that gene to place individuals, and any other data that you might have on that turtle or other turtles are irrelevant in terms of placing that animal back on the landscape. Okay, it's not working. There we go. With alternative 2, where for the reference animals you use the whole genome, one of the great powers of that is that then any data you collect on the animals you want to repatriate or you want to learn about can be compared back to the reference genome, the reference animals, because for them you have the whole genome. So if, for example, you had one study with JJ's markers, which are based on a sequence capture probe, and you had another set of animals which were based on Peter's RAD-seq data, either of those can be compared back to those reference animals. It makes it a more kind of powerful thing. And in addition, if you had, say, old animals that are no longer around, but where someone did some old school Sanger sequencing on them, you could, in retrospect, map those back to those reference animals as well. Any genetic data you have, you can map them back. So how might this work? Well, here's just one example from a project Peter and I are involved in right now. So the map on the left shows Edwards Air Force Base. It's sitting out in the middle of the Mojave Desert. Not great turtle habitat, but there is some wetlands out there. And much to everybody's surprise, some consultants picked up some turtles, some Western pond turtles from out there. And the only thing, Peter and I went out and helped catch some turtles and took tissue samples from them. And we're in the process of working those up right now. The only thing we know about them at present is that they have this enlarged inguinal scute, which is a sort of taxonomically important character. So for a different project, one of the things Peter and I are doing is taking a collection of about 150 turtles, Western pond turtles from across the state of California. And we're resequencing them. So those are sort of our red dots, if you will, on this map. We're sequencing them for their entire genome, okay? And we can then use that as a reference set, and we're in the middle of doing that work right now, to compare to the Edwards Air Force Base animal shown here in red. And the fact is that that animal could be a part of the Sierra Nevada population, which is relatively close by, of Emmys or Actinemys marmorata. It could be a part of the also nearby coastal clade of coastal Emmys or Actinemys pallida, or they could be released from anywhere. And we should be able to figure that out with fairly fine resolution using this data set that we're putting together. Everyone always wonders about cost, and that's a legitimate question. If you're doing this where you want to have the reference animals be whole genome resequenced organisms, there's two components to that. The first is a single gold standard genomic resource. This is kind of like the turtle Peter was talking about, only done at even a better level, at a chromosome level resolution. And it's a really important resource to have. And those right now, with today's technology, run about $8,000 for a 2.5 to 3 gigabase turtle. Resequencing those reference animals for having nice, complete whole genomes, as Peter said, you don't need to redo the whole thing. It's a much simpler exercise. Those can be done for about $250 a pop. So for our study with establishing that resource for the Western pond turtle, it's about $37,500 for the resequencing for those 150 animals, $8,000 for the reference. It's about $45,000 to do the whole thing. Not cheap, but not super expensive either. Okay, sorry. So if we consider one of JJ's prime targets, also one of mine, Clemmys guttata, the most widely confiscated turtle species at LAX, at least as far as Joe Ventura has told me, it occurs in 21 states plus one Canadian province. If we thought about a level of resolution of, say, 5 per state, say maybe 100 turtles, that cost would be about $33,000. And if we can use the reference genome from the Western pond turtle that were developed, that gold standard turtle, which we probably can in this case, that would reduce the cost down to about $25,000. Again, kind of getting within reach. And what I just want to say here is that we can do this. This is something that we're doing it right now for Western pond turtles. It's something that we can do. We need tissues. We do need some funding. We need to establish pipelines for geolocating those samples, putting them back out on the reference landscape. And we're developing those pipelines, which will hopefully be generalizable right now for the Western pond turtle. And we need a stable web presence to support some of this reference material. And I'm thinking the TSA might be a good home for that. If you're interested, remember, we can pull together tissues right now, as JJ suggested, and we can try a pilot. And if you're interested in that, I encourage you, please get in touch with me by email. There's my cell phone number. Give me a call and let's talk about this. Okay, that's what I have to say. Quick thanks to Tim Gregory for financial support, to a bunch of people from our lab over the years, including Natalia Gallego Garcia, who's in the lab right now, and to different funding sources. And I will leave it at that